Hello everyone, and welcome back to another thrilling discussion in anatomy and physiology, the second half of the unit. So today we're going to be talking about blood vessels and circulation. So without further ado, let's just get this thing moving. So blood vessels are the conductors of the blood that we studied in chapter 19 to the various places in your body. And because your body is such a vast, diverse universe, there are different types of blood vessels. The largest blood vessels are going to be found in the following places. They're going to attach to the heart, and that's pretty intuitive and pretty straightforward because your heart's job is to pump blood through these conducting vessels to the various parts of your body, so that makes sense. And then you also have them attached to your pulmonary trunk, and then you have your aorta. So the largest blood vessels, aorta, pulmonary trunk, they're going to have the attachment to the heart. And what we know about the pulmonary trunk is that we have blood that leaves out of the right side of the heart and goes through those pulmonary arteries to the lungs to pick up oxygen, and then goes back into the left and right pulmonary veins to go back into the left side of the heart, where then it's conducted out of the aorta to the various parts of the body. The smallest blood vessels we call capillaries. And they're also called resistance vessels as well because they're so small. In fact, they're actually microscopic in a lot of ways, that they resist the flow of blood. So the flow of blood doesn't go through them as quickly as you might find for those larger blood vessels or even medium-sized blood vessels. But the smallest blood vessels are capillaries, and they're going to be found pretty much anywhere that is vascular, any place that there is a blood flow. Capillaries, as I said before, they're microscopic, and they have a very, very thin wall. And in fact, of the three layers that may be present in blood vessels that we'll discuss in a moment here, capillaries just only have one layer. They have the endothelium, which is just simple squamous epithelial cells. So that's just that one layer through there. So these are our exchange vessels. They're able to exchange the oxygen for carbon dioxide and also to exchange nutrients through the wall because they are so small and they're pretty widespread. So the different layers of a blood vessel that they can have, we call those tunicas or tunics. So tunic layer from the Greek or the Latin. I don't really remember which one. So blood vessels can have up to 3 layers to them. They don't have to all have 3 layers, but they can have up to 3. The tunica intima is the internal layer, also known as the endothelium, and that's the innermost layer of the blood vessel. The more muscular layer of the blood vessel, where those smooth vessels will be found, is going to be in the middle layer, what we call the tunica media. And then we have the external layer of the blood vessels called the tunica externa. And many times that tunica externa's job is just to help anchor the blood vessels in its place so it doesn't move too far away. So when we talk about blood vessels, you've probably already recognized this, and you've probably seen it in lab class already, that there are differences between an artery and a vein, and not just because one is blue and one is red, not just because, you know, that's not the only thing. And we recognize that the blue and the red, they're going to directly correspond to oxygen carrying capacity. So that's what those colors stand for. So a couple of things to make a special note of is that for your arteries and their veins, you're going to find them pretty much lie side by side. So where there's an artery, you'll find a vein. And we saw that in our heart models, that we saw an artery and we saw a corresponding vein that went with it most of the time. We understand that arteries are going to be thicker, in the walls of them are going to be much thicker, and they're going to be able to tolerate a higher blood pressure. And from what we understand about the heart, that also makes sense because it's able to withstand all of that pressure that the heart is exerting on the blood that's moving out of the aorta and then out of the various parts of the body. That pressure is going to be higher in the aortas than it's going to be in your return system, which is your venous system. A collapsed artery has a small round lumen, and remember, a lumen is just the internal space, whereas a vein has a more of a large flattened type of lumen. So veins look a little bit more floppy. They're not nearly as rigid as what we would expect for the arteries because they don't really have to withstand such high pressure. the lining of the vein is going to contract, and that's meant to help facilitate the movement of blood back to the heart. And whereas the arteries, they don't have a lining that really contracts. It doesn't do that. Artery lining can fold, but it won't contract. Arteries are much more elastic, and that plays right into the fact that it has to be able to withstand much higher blood pressure than the veins do. And then to compensate for the fact that veins are larger, floppier, and they don't really have as high a pressure of the blood moving through them, veins are going to have valves in order to allow for that blood to move back up towards the heart. So first one, we're going to talk about our arteries. So remember we said just a moment ago that arteries were much more elastic, and it allowed them to be able to withstand really high blood pressure when the left ventricle of the heart was pumping forcefully all that blood out of the aorta. So the elasticity allows it to withstand the pressure that comes with each heartbeat. The contractility of the arteries, how much those smooth muscles are able to contract and then also to relax. Constriction of those blood vessels is known as vasoconstriction, and that's the contraction of the arterial smooth muscles. And then the dilation, or the relaxation of them where they're getting larger, that's called vasodilation. Now, what controls whether or not these muscles contract or they relax is not going to be your conscious side of your nervous system, if you will. It's going to be more of your non-conscious, so what you don't have to consciously think about. Like for your smooth muscle, those are under your conscious control. You tell your arm to relax, you tell your arm to pick up, you tell your leg to move out, you scratch yourself, you move your neck, whatever it is that you are consciously telling yourself. Your autonomic nervous system, on the other hand, doesn't quite work that way, remember? Your autonomic nervous system is going to have information that's sent through through the sympathetic division, and it just tells it to do it. So it's like your heart beating, breathing, all of those things you don't have to consciously think about. And the same thing with vasoconstriction and vasodilation. There are nerves, yes, that come from your central nervous system, but you don't have to consciously think about those things happening. It's part of your autonomic nervous system. So as we go from the heart to the capillaries, the arteries are going to change size, and they're going to change a little bit of their function. So we'll go from those elastic arteries, the closer that you are to the heart, so the aorta is a very prominent elastic artery because it has to be able to withstand all of that blood pressure that's coming out of that heart and be able to expand and then recoil back. Then we have muscular arteries as we get a little bit further away from the heart, and then we have arterioles, which are going to be much smaller in diameter, and they're known as kind of resistance or exchange vessels. So the arterioles are really small. So they're much smarter than just your muscular arteries, and they're definitely much smarter than your elastic arteries. They don't really have much of an external layer. So remember those 3 layers we talked about that a blood vessel could possibly have? The arterioles, they don't really have that tunica externa. They do have a little bit of a tunica media, so there's some smooth muscle there, but it's really not a complete layer of it. It's not all joined together, because as you get closer to the capillary end of the arterioles, then you're going to see a lot less of that smooth muscle. So closer to the muscular arteries, you'll see more of this tunica media, but as you get further away down to those exchange vessels, of the capillaries, you don't see it as much. So that's why it says can have either a very thin or just an incomplete layer of tunica media. For small muscular arteries and the arterioles, they're going to be much smaller in diameter than what we have for the elastic vessels, so they're going to resist blood flow. So that's why we can call these resistance vessels. And sometimes you'll hear your book, or even if you're reading your book, you'll hear them referred to as resistance vessels because they are so small and they're going to resist blood flow, even more than that of the capillaries, or not as much as the capillaries. So, but even more than the elastic arteries and those muscular arteries. So the arterioles are known as the resistance vessels. So issues that we can have with a blood vessel, in arteries in particular is an aneurysm. And an aneurysm is just a bulge in the arterial wall that does not necessarily always equate to a break. If you have an aneurysm that happens in different parts of your body, your brain quite notably, then you will find that that can be quite fatal very quickly. Usually it's some weak spot in those elastic fibers, so the blood pressure that's going through there, those arteries can't withstand that blood pressure, and so it causes it to kind of bulge out. Or you can have the collagen fibers that make up some aspects of the blood vessel to help kind of maintain the integrity of the wall. If the collagen fibers are not developed as robustly as we would like them to be developed, as we see in people that have Marfan's condition, then that can also cause weak spots on the elastic artery, and it can cause the vessel to rupture. So here we have some videos, or some links to videos that discuss an aneurysm a bit more. I won't be able to link it through here. It just doesn't, the Collaborate session doesn't work that well for showing those videos. It's really difficult to hear. But I do invite you to look these videos up. So going down to the smallest of your blood vessels, you have your capillaries. And the capillaries, they can be called resistance vessels, but because they do resist the flow of blood. In fact, they resist it so much that the blood cells that go through there have to go in single stacks that we call rouleaux. The capillaries are the smallest of the blood vessels and they have the thinnest wall. So they completely lack a tunica externa and a tunica media. They're just tunica intima. In fact, as I said before, they're really just microscopic networks of these tissues that allow for nutrients and waste products to be exchanged through the blood flow. So they diffuse blood between the blood and the interstitial fluid. So there are three different types of capillaries that we're going to talk about. We can have continuous capillaries. That endothelial lining they have, that tunica intima, that's going to have a complete layer of tunica intima. Where we find continuous capillaries, that they're pretty pervasive. They're pretty much everywhere in most tissues except for the epithelium and the cartilage, which for us makes sense because the epithelia and the cartilage, they are avascular tissues. The function of continuous capillaries, now this is pretty important here, so we're going to highlight this function, what its job is. Remember in A&P2, and especially in the lecture, we spend primarily our discussion is not towards anatomy, but it's more towards physiology, so the function of things is always very important. It's able to permit the diffusion of water, small solids, and lipid-soluble materials, and also to block blood cells and plasma proteins from leaving out. So those continuous capillaries, they're meant to just exchange nutrients for waste products from that interstitial fluid. They're not going to really let anything go out of it. Larger things like blood cells and those plasma proteins, when they're in continuous capillaries, we want to keep them in. So we do have some specialized continuous capillaries that we find in places like the central nervous system and the thymus. For the central nervous system, we've talked about how we've had those neuroglial cells that their job is to maintain that blood-brain barrier. These specialized capillaries have a tendency to be even more restrictive. And as I said before, when we talk about that blood-brain barrier, that's an example of specialized continuous capillaries, that there's an additional layer of protection to keep certain things from getting into the interstitial tissue of various organs. So the next class of capillaries that we're going to talk about are fenestrated capillaries. And fenestra is a window. So these are going to have windows, if you will, in the capillary, or another way to say that is pores. They have little pores that are in there. So the endothelial lining is not complete. And when I say it's not complete, there are going to be little pores that happen. And because we have those pores in these fenestrated capillaries, what it will allow for is the rapid exchange of water and larger solutes between the plasma of the blood and the interstitial fluid. So larger things can get through here, and this is all by design. Places that we find fenestrated capillaries are going to be in endocrine organs because there are hormones you want to get in and out of circulation in the bloodstream. Also in your kidneys, your job of your kidneys is basically to act as a filtering station. So there are some things that we want to filter into the filtrate or the urine, what's going to become the urine, and other things that we want to filter out and put back into circulation. We'll have a whole discussion on this in the urinary chapter. And then even the intestinal tract, much the same way as your kidneys, there are some things that we want to keep, those very large molecules, those solids of the foods that you've eaten. We want to keep those things and put them in circulation, where there is some stuff that it should just be filtered out as a waste product and make its way through the digestive system and right on out your body. So these fenestrated capillaries, the locations in which they're found, are meant to help facilitate the rapid exchange of molecules like water, and also to facilitate the exchange of molecules that are just much bigger and they're too big to just diffuse across the plasma membrane of those simple squamous epithelial cells that we find in the endothelial lining of blood vessels. And then our last class of capillaries here is that we have sinusoidal capillaries. So fenestrated capillaries allow for the rapid exchange of things because we have those little pores that are embedded within the capillary wall itself. Then sinusoidal capillaries are going to really allow for a much greater exchange. These are actually going to be gaps, if you will, or holes between the cells. So the cells that make up the blood vessel are going to have these large areas. And in your textbook, it shows you a really neat picture or a drawing of these sinusoidal capillaries. So places that we find them would be the liver, the spleen, the bone marrow, and even more so here, or as much here, we have in the endocrine organs. So the job of the sinusoidal capillaries is to promote pretty much a free exchange. So we still have things like water and larger solids going through as we had in the sinusoidal capillaries, but we're also going to have some plasma proteins that are going to get a chance to get out of here as well. And we just have a much more freer exchange. Now in places like your bone marrow where we make our blood cells, that makes sense that we have sinusoidal capillaries there because we need the blood cells to be able to get in and out of circulation, those reticulocytes once they've been made. Because there is such a rapid exchange of material and then such a rapid exchange of lots of bulk material that we have from these sinusoidal capillaries, it makes sense that these areas are going to be heavily monitored by phagocytotic cells because we don't want anything to get into circulation that shouldn't be getting into circulation. So to kind of keep things where they need to be, we have phagocytotic cells that will monitor the blood and what's going out through these really large areas. So here we have a picture of a continuous capillary, and this continuous capillary on here, we see the boundary between the cells, but we don't really see a space at all. We don't see any pores or anything. And these little dots, these are the endosomes, so they're just structures within the cell. Whereas if we look at a fenestrated capillary, we still see the boundary between the epithelial cells, but we also see these little pores here. So we see these windows or fenestrations. And then for the sinusoidal capillaries, notice that there's a huge, huge comparatively speaking between the fenestrated and the continuous capillary. These huge gaps between the cells that will allow for the free movement of various different types of molecules. And on the outside of this area, and even on the inside of the cell, pretty much on the outside, you're going to find those phagocytotic cells monitoring that area. So what we call a capillary bed is a plexus, and it's just a network of capillaries that will have at one end the arterial end, and then at the other end the venous end. And within that plexus or that bed, you're going to have the exchange of nutrients for waste, and then once that exchange has taken place, and oxygen for, carbon dioxide for oxygen, then those deoxygenated cells and the waste product, wherever it's carrying, is going to make its way through the venule end. And the venules are like the arterioles in that they are the smallest of the veins, and they'll make their way back up to the heart. So what allows for the blood to move through the capillaries in a more controlled type of fashion is that we have these precapillary sphincters that are going to guard the entrance to the capillary. So when it opens and closes, it causes the blood to flow in pulses. So it's not that these sphincters are constantly closed or constantly open. They're contracting and relaxing and contracting and relaxing to allow for blood to flow through there in pulses. And this will allow for the exchange of materials like oxygen and carbon dioxide, nutrients for waste products to happen in an efficient manner. So here we have a beautiful picture of a capillary bed or a capillary plexus here. The red end, that's going to be the arteriole, so the oxygenated blood would be flowing through here. The arterial end of it, you can see we have some smooth muscle cells around there, but then once we get down here to the capillary bed, we don't have the smooth muscle that's just completely found all around it. We just have these areas that may have these sphincters around it. So you have these precapillary sphincters, these areas right here, and they would contract and relax to allow the blood to flow through in a more controlled fashion. Now we do have down here an anastomosis where it's the arterial venule, so it's just kind of a variable blood flow that will go through here. So angiogenesis is just known as the formation of new blood vessels. And we see angiogenesis take place a lot of times when the tissue in that area is being starved or deprived of oxygen. So we'll have some angiogenesis take place. We can see angiogenesis also take place in places that we don't want, and that would be, an example of that would be for cancer cells. Sometimes cancer cells can exhibit or have some of this growth factor, which is called vascular endothelial growth factor, and it will allow for blood vessels to form in cancerous cells, and we don't want that to happen. Angiogenesis is something that should happen primarily in the embryo because these tissues are growing and developing. But once again, as I said before, we can have some of this growth factor released when the tissues are in a hypoxic environment or starved of oxygen. This is most important in cardiac muscle, as we see that in patients that have chronically constricted or occluded or blocked vessels. If we start to see some angiogenesis take place, that lets us, we see that and we know that this is probably something that's been going on a long time because those tissues are starved of oxygen. So they're like, oh, we need to build more superhighways to get this oxygen to this area. Whether it's in cancer or a person that has chronically constricted or blocked vessels, neither one of those cases are points when we want angiogenesis to take place. Angiogenesis, we really should just see it happening in embryonic development. Vasomotion is the movement of blood through these blood vessels. Contraction and relaxation cycles of these capillary sphincters allows for this vasomotion to take place. Keep in mind that the further away you get from the heart, the less pressure do we have generated. So when we get down to these capillary beds, we don't really have a whole lot of pressure that's happening through there. And especially when we get into the venule ends, we're not going to have as much pressure, so there needs to be some structures in place that will allow for the blood to still make its way back up to the heart, even if it's down there in the pinky toe and it has to fight gravity and the lack of pressure there. But the vasomotion and the role of those capillary sphincters are going to allow blood to flow constantly through that area. Now, however, what areas are going to be allowed blood access to, that can change. So the capillary beds are constantly changing the routes and the pathways of that blood, and that is a good thing because it allows for all of the tissue in the place where that capillary bed is found to have an adequate supply of oxygen. So now moving on to the veins. The veins' job is to return blood to the heart, which you've learned from the previous chapter. Collects blood from the capillaries and the tissues and the organs, returns it to the heart. Much larger diameter than arteries are. They have much thinner walls and they're a little floppier. And as I said before, and I've been kind of saying this repeatedly, they're going to have a lower blood pressure because remember, the further you get away from the heart, going from the elastic arteries to the muscular arteries, to the arterioles, to the capillary beds, and then from the capillaries onto the venules, to the medium-sized veins, to the large-sized veins. The further you get away from the heart, the lower that pressure will be. So the venules are the counterpart to the arterioles, which are the smallest veins, and they'll collect blood from the capillaries. If the arterioles are going to deposit blood into the capillaries, the arterioles collect it from it. And then we have the medium-sized veins that have a thinner tunica media because they don't have as many smooth muscle cells, so they don't have they're not as muscular as the arteries are. And remember, this is just something that we've already established and talked about in the very first slide that we went over. Large veins have all three layers, externa, media, and intima. Those veins also have valves, and this is something I'm going to highlight for us because it's important. What those valves allow the veins to do is it allows it to compensate for the fact that it has a lower pressure in the veins, and it helps to make sure that the blood that goes back to the heart doesn't flow backwards. So they're folds of the tunica intima, or that innermost endothelial layer of the blood vessel of the veins, and it prevents blood from moving backwards. And the compression of your muscles outside of your veins helps to further push blood towards the heart. And once again, this is all done in an effort to compensate for the reduction in pressure, and also to combat gravity to get blood back up to the heart. So here we have a beautiful picture of this whole thing happening here, that when the muscle is relaxed, we have the vein is closed, but when we contract the muscle, that valve, so we have contraction here, that valve opens up, and then the portion of the muscle that's not contracted, it will be relaxed, and when it's relaxed, that vein is closed. So this just ensures that the blood moves up in one direction, back towards the heart. So, another thing I wanted to kind of discuss there. So if you've ever talked about or heard about varicose veins, so what's happening with the varicose veins is that they are veins that are not, the compression of the muscles is not enough to prevent the backflow of blood, so the blood will just kind of pull out in those areas. So with a varicose vein, the valves are somewhat damaged, if you will, and they are going to be, instead of being nice, straight, and taut, they kind of just leak over, and the blood doesn't go through there as nicely, go through those valves as nicely. So that's why compression socks and compression hose, that sort of thing, are helpful to overcome that. And there is surgery that you can have for it if needed. So now on to blood pressure and resistance. So some terms before we start to talk about this blood pressure and resistance. Blood pressure, we measure it in millimeters of mercury, because there's a lot of ways that you can measure pressure in atmospheric pressures and ATM. You can measure it in torr, but we're going to use millimeters of mercury. So the blood pressure, what we're really measuring the pressure of is the arterial blood pressure, because remember, the higher of the two blood vessels that we have pressure in are in the arteries. Capillary hydrostatic pressure is going to be pressure within the capillary beds, and then venous pressure is pressure within the venous system. Typically, we don't talk about a lot of venous pressure because it is already just so much lower. Turbulence is a swirling action that disturbs the smooth flow of a liquid. In this case, it would be blood. And we find turbulence that happens in those large chambers of your heart, so the right and the left atria and the right and the left ventricles. We also have turbulence that happens in those great blood vessels, so the aorta and the pulmonary trunk. When we have arteriosclerotic plaques, can cause abnormal turbulence, where we can have little bumps, if you will, in the blood vessels where we have plaque that's building up, that's going to just cause the blood to swirl and have a disruptive flow when it shouldn't. So here's a really cool picture that's kind of showing how blood pressure changes as we go from the aorta down to the vena cava. So that's coming back to the heart, the inferior and the superior vena cava. So we have the blood pressure is about 100 millimeters of mercury when we leave the aorta. By the time we get down to the venules and the veins, it's much lower. It's lower than even half of that. So for that reason, to make sure that the blood efficiently gets back to the heart, we have the valves in the veins, and we also have something called the respiratory pump, which we will talk about in just a second. Now, the velocity of that blood flow, although the velocity of the blood flow, as we would expect, is very high at the aorta because you have the pressure, it's got much higher pressure from the left ventricle of the heart forcefully contracting it. Much slower velocity, it's not moving as fast through the capillary beds, but we see that it starts to kind of pick up momentum or pick up velocity as we start to go back to the heart. And one of the reasons for that, once again, is this respiratory pump and also the fact that these veins have valves. And muscular contractions help those veins to help the blood to continue to move in one direction, help the blood to move, and the veins keep it from moving in an opposite direction. So what normal blood pressure is 120/80. Anything greater than 140/90 is considered hypertension, and that's high blood pressure. And then low blood pressure is just abnormally low blood pressure, anything lower than 120/80, which for some people like myself, my blood pressure is like consistently like 95 or 100/60. And it's for the biggest issue with hypotension is you can have dizzy spells, fainting, vertigo, because you're not getting enough blood up to your brain. If you're usually a 120/80 person, then having anything like my pressure would be too low for you. I think they're even saying now, and some hospitals and some cardiovascular specialists are saying that even 120/80 might be starting to get a little too high. So I know there's been some debate about that. But if you just already have low blood pressure, then it's not really an issue. Typically, if you have a normal blood pressure and then it's low, that can usually result in vertigo or dizzy spells. So the pressure in the small arteries and the arterioles, we understand that it's going to change. What the mean arterial pressure and pulse pressures are going to decrease as you get further away from the heart, which we saw in our in the previous two graphs, and then we also talked about in the beginning of this kind of lecture. Blood pressure decreases with friction, and pulse pressure decreases due to elastic rebound. So with each one of the beats of the heart, that pressure that's exerted on the walls of those blood vessels is going to become less and less, becomes less because of the elastic rebound, the ability of those blood vessels to, those elastic blood vessels to take that really hard forceful contraction and pumping of blood through there and to be able to absorb that. So venous pressure and venous return, the muscular compressions of peripheral veins, and also those one-way valves help to push that blood up in one direction. And then we also have the respiratory pump, and we'll talk about this again in the respiratory chapter. But what the respiratory pump does is it allows to, there's going to be a change in pressure between your thoracic cavity and your abdominal cavity, because really we only have trouble getting blood back to the heart when we're going from those areas below the heart because of gravity, right? That makes sense. So when we're trying to get that blood back up from, let's say, your pinky toe, we have the fact that veins have valves. We have the contractions of muscles and those one-way valves to help move things back up. But what also helps to increase the velocity of that blood flow, because remember, it also starts to heat back up and increase as we leave the capillary beds, is the action of this respiratory pump. So when you're taking inhalations and exhalations, the pressure in your thoracic chamber where your lungs are found or in your chest is going to be different on inhalation and exhalation. When you inhale, that's going to decrease your thoracic pressure. So everyone take a deep breath in. When you take a deep breath in, what you're doing is you're filling your lungs with oxygen. Pressure and volume are inversely related to one another. As pressure increases, volume decreases. Okay, fair enough. As you filled your lungs with oxygen, the volume increased, so the pressure is going to decrease. That's Boyle's law. Pressure and volume are inversely related to one another. They're inversely proportional. So when you take a deep breath and you expand your lungs, you have less pressure that's in that area, in your thoracic area, so that means the pressure that's in your abdominal area is going to be much higher. So because you now have this higher pressure, what it can do is it helps you pull that blood up through your through the veins and increases the velocity of it. Just that change in pressure on the outside of the blood vessels is enough to suck it on up. When you exhale, we've reduced the volume of air in your lungs, you reduced your lungs, and so the pressure outside of them is going to increase. So it has a raise of thoracic pressure. So that's how that respiratory pump works, and that it's just a change in pressures in your thoracic cavity that will allow for the blood to be pulled upward. And we'll talk about the respiratory pump again in the respiratory chapter. All right, so this is where we're going to end for the moment for this session, and then we'll come back with the part 2 of the blood vessels. I hope you're having a great morning, noon, or night.